Scientists said the average human brain may contain enough microplastic to make a disposable spoon. Then other researchers noticed something disturbing. The machine may have been measuring the brain itself. But in 2026, a new study found plastic in brain tissue using different methods and samples taken from living patients. So the plastic may be real, the spoonful may not be. And the difference between those two statements is where this story becomes much stranger. In February 2025, researchers published an analysis of human liver, kidney, and brain tissue. The brain results were unlike anything they expected. Samples collected in 2024 produced a median estimate of nearly 5 milligrams of microplastics and nanoplastics per gram of tissue. That was 7 to 30 times more than the liver or kidneys, and roughly 50% higher than brain samples collected in 2016. Extend that concentration across an average adult brain, and the result is close to 7 grams, a small plastic spoon. The comparison escaped the paper almost immediately. Plastic was no longer only in the ocean, the air, the food chain, and the blood. It was supposedly accumulating inside the organ, currently deciding whether to keep watching this video. The study found mostly polyethylene, the material used in plastic bags, films, containers, and bottles. It also reported even higher measurements in 12 brains from people diagnosed with dementia. The perfect headline had arrived. Your brain is full of plastic. The amount is increasing, and people with dementia may have even more. There was only one problem. The researchers never removed 7 grams of plastic from a human brain. They never collected a spoonful of tiny particles and placed it on a scale. They tested small pieces of frontal cortex. Then they used the concentration found in those samples to estimate what might exist across the entire organ. The spoon was already an extrapolation, and the original concentration depended on a machine correctly separating plastic from one of the most chemically complicated tissues in the human body. Most suspected particles were too small to count reliably with an ordinary microscope. Many appeared to be nanoplastics, some shorter than 200 nanometers. So the researchers mainly used a technique called pyrolysis gas chromatography and mass spectrometry. The process sounds complicated, but its logic is brutal. First, chemicals digest as much biological tissue as possible. Then the remaining material is heated until it breaks apart. The gases released during that destruction are separated and analyzed. Different plastics produce characteristic chemical fragments. Detect those fragments, and the machine can estimate which polymers were present and how much material created them. It is not like photographing a suspect. It is like burning the entire crime scene and identifying the suspect from the smoke. That can work extremely well, but only when nothing else produces similar smoke. The human brain is rich in lipids. These fatty molecules form cell membranes, carry signals, and insulate nerve fibers. When biological lipids and other organic compounds are heated, some can break into hydrocarbon fragments. Polyethylene also breaks into hydrocarbon fragments. This means that if the tissue is not removed completely, normal brain chemistry may contribute to a signal interpreted as plastic. The machine does not see a shopping bag. It sees molecular wreckage and works backward. The original team knew about this risk. They treated the samples with potassium hydroxide to destroy proteins, fats, and other tissue before analysis. The treatment reduced liver and kidney samples by more than 99%. The brain behaved differently. From 500 milligrams of starting tissue, the liver and kidney left an average of about 3 milligrams of solid material. The brain left approximately 41 milligrams. Something had survived the digestion. The researchers argued that this was consistent with the brain containing much more plastic, but they also acknowledged that unknown organic material probably remained and could influence the result. That creates an uncomfortable loop. The brain left more residue because it contained more plastic, and the brain was estimated to contain more plastic because that residue produced a stronger plastic signal. Both statements may be true, but one cannot automatically prove the other. A separate team tested the same type of analysis using human blood. They added known amounts of several plastics and asked whether the method could recover them correctly. For some polymers, it performed reasonably well. For polyethylene, it did not. Biological material created interfering signals and non-specific breakdown products. The researchers concluded that the method, in its current form, was not suitable for reliably measuring polyethylene and polyvinyl chloride in biological samples. That mattered because polyethylene represented most of the plastic reported in the brain study. The possible interference did not attack a minor detail. It attacked the polymer responsible for most of the spoon. In late 2025, scientists formally challenged the original result. They questioned whether brain tissue had been removed completely, whether contamination controls were sufficient, and whether phospholipids and other natural components could imitate polyethylene. The images did not end the argument. Electron microscopy showed tiny carbon-rich flakes and shards. Plastic is rich in carbon. Unfortunately, brain tissue is too. The team had stronger evidence than ambiguous pictures. Infrared testing supported polyethylene in 5 samples. Some material was analyzed in a second laboratory, and contamination controls were used. The older samples had also spent much longer in storage, yet the newer brains produced higher readings, so the study could not be dismissed as simple contamination. But its methods still proved different things. Imaging supported the presence of particles. Pyrolysis produced the enormous mass estimate. The particles could be real, while the spoonful was wrong. Then the story changed again. In April 2026, a different research team examined 156 brain samples from 113 patients undergoing surgery for brain tumors. They also tested 35 healthy brain samples from 5 people who had recently died. Many samples came directly from living patients rather than old tissue banks. The team used several methods, including infrared-based imaging, capable of chemically identifying particles. It also recreated parts of the surgical process and tested possible contamination sources. Microplastics and nanoplastics were reported in almost every diseased sample and every healthy sample examined. Contamination can never be eliminated perfectly when plastic exists throughout hospitals, laboratories, equipment, clothing, and air. But the entire phenomenon probably was not created by one machine analyzing old tissue. Plastic appears to be reaching the human brain. That is the first major correction to the skeptical version of this story. The second is just as important. The new study did not confirm the 7 grams. Most living samples came from tumors or tissue surrounding tumors, where the blood-brain barrier may already have been damaged. The healthy comparison group came from only 5 donors, and methods that identify individual visible particles do not necessarily measure the total mass of the smallest nanoplastics accurately. One method can be better at proving that particles exist. Another can be more sensitive when estimating total mass. Neither automatically gives a perfect answer to both questions. So 2 statements that sound contradictory may both be correct. There is probably microplastic and nanoplastic inside human brain tissue. We still do not know whether an average brain contains anything close to 7 grams. We also do not know whether those particles are causing disease. The original study found higher measurements in brains from people with dementia. The 2026 study found more plastic around brain tumors than in its small, healthy group, and reported a correlation between plastic surface area and tumor proliferation. Neither study proved that plastic caused dementia or cancer. Dementia can shrink tissue, weaken the blood-brain barrier, and interfere with the brain's waste clearance systems. Tumors also damage barriers and transform the tissue around them. Plastic could contribute to disease. Disease could make plastic easier to accumulate. Both could be influenced by something else. Or, part of the apparent connection could still come from measurement. Finding more firefighters at larger fires does not prove that firefighters started them. Cell and animal experiments provide reasons for concern. Under certain conditions, these particles can trigger inflammation, cellular injury, and changes in blood flow. But laboratory animals often receive manufactured particles at controlled doses. That is not the same exposure as a person inhaling tire dust, swallowing synthetic fibers, and drinking from plastic containers for decades. The dose, shape, and chemistry all matter. Plastic pollution is real. Humans inhale and swallow tiny plastic particles. Multiple studies now support their presence in human tissues, including the brain. But the most frightening version of the claim traveled much further than the evidence. Scientists did not find a spoon inside anyone's skull. They measured a concentration in small samples, then extended it across an entire organ. That estimate came mostly from polyethylene. Polyethylene is also one of the hardest polymers to distinguish from biological material using this type of analysis. The original study produced supporting evidence. Its critics identified real weaknesses. A newer study strengthened the case that particles are present without confirming the viral mass estimate. So, is your brain full of plastic? It probably contains some. It may contain more than several other organs. The amount may be rising as environmental exposure increases. But nobody can currently say with confidence that half of 1% of your brain is plastic, that you are carrying 7 grams of it, or that it is causing dementia or cancer. The particles appear real. The quantity remains disputed. The health consequences remain unresolved. That difference matters because once a frightening estimate becomes an object people can imagine, the object replaces the experiment. A chemical signal becomes 7 grams. 7 grams becomes a spoon. The spoon becomes something scientists physically removed from a human skull. Each step feels small. By the end, a disputed measurement has transformed into an event that never happened. Future methods may confirm the original estimate. They may find even more plastic. Or they may show that part of the most alarming signal came from the biological material the experiment was trying to remove. For now, the strongest honest conclusion is stranger than either headline. Plastic has probably reached the human brain. But the spoonful may exist only in the way we measured it. The plastic appears to be real. The spoon was always a model. And between those two statements, the internet discarded almost the entire story.